Hi guys, welcome to the Sunday session. Just give me a quick thumbs up or emoji or something if you can hear me, just to let me know I've got this set up correctly. Hopefully there was a little bit of a problem getting all set up today, so I'm hoping it's working. So yeah, if you could let me know, that'd be great. It's saying for me the chat isn't working. Are you guys having any more luck? I can't, it's not loading for me. Chat disconnected. Let me try. I'm just going to try refreshing it. Classic. Let's see if that works at all. Doesn't want to do that. Oh, Elena, perfect. Thanks for that. Yeah, my chat's still being funny. I think this happened before, like a month ago. I had to keep refreshing it the whole way through. I don't know why it's like that, but we can work with that. I'll keep refreshing it. It should be the same for you guys, so that's not too much of a problem. These, Tony, yes, these happen. So it's once a month for each subject. They're not always the same time in the month though. So the next biology one, I'm actually not sure. You can, if you look on the actual page, you can see all the calendar of all the different classes, and they also do ones in chemistry, maths, and physics. So if you do any of those subjects, you can join for those as well. Anyway, yeah, let's make a start today because we're already a few minutes late. So we're looking at mitosis today, a really important topic in the AS syllabus. So you usually learn this alongside meiosis, but we're just going to focus on the mitosis part today. So we'll go through the different bits. We'll do some past paper questions, same as normal. These sessions, obviously, we're running them live, so they're meant to be interactive. So I'm going to ask you guys questions throughout the session, and yeah, feel free to put an answer in the chat. It works better, the sessions, if you interact with them, and generally people find they get more out of the session if they're interacting with it. It's totally optional, but yeah, it can be a good way because I can look at your answers as well and pick up on any mistakes and stuff in them. So yeah, a little bit about me. My name's Alex. I'm the head biology teacher here at Snapprovise. I've been tutoring and teaching biology for the last 5 or 6 years now. And yeah, we normally have a page with the aims for the session. So the aims here today is just to go through the mechanism for mitosis, go through the different stages, look at the cell cycle as well, and then we'll do some past paper questions just to make sure that you guys are all up to speed with everything. So we usually start with a bit of revision. This can be from different topics that relate to the one we're going to look at today. So they could be relating to mitosis or just random things to just to help you guys cement different ideas. So this is a throwback right to the start of AS. Most people do microscopy at the beginning. So yeah, how would you tell the difference between an SEM and a TEM micrograph? And if you're not sure what a micrograph is, it's just the picture you get from a microscope image. Let me turn my phone on to do not disturb. It's buzzing. So yeah, I'm gonna have to remember to refresh the page to get your answers. Nice, yeah. The two are 3 dimensions. So these would be 3D images. and these would be just in 2 dimensions, 2D images. Cool, nice, thanks, Tony. So next one, how is prokaryotic and eukaryotic DNA different? And there's probably quite a few things you can put here. What can you guys come up with? And yeah, I just had some answers come through now for the first one. It's gonna be it's gonna be a bit of a struggle with this. There's like a 10-second delay at least. Yes, TEM, you can see internal organelles, very good. SEM, because it's an electron microscope, you actually can see organelles, but the resolution of TEM is several orders of magnitude higher than a scanning electron microscope. Diksha, actually, no, neither of those have color. They're both like black and white. Anytime you use an electron microscope, you don't see real color. You only see it in black and white. But for both of them, you can add color back afterwards. So it's like artificially added on a computer. But you can do that for both. So that actually wouldn't be a suitable way to tell them apart. Cool. And then what you guys come up with for the DNA? Yeah. Okay, let's work on this one now. So unicellular, multicellular, that is true, but that is not related to the DNA. So I wouldn't put that. I mean, I guess like, yeah, unicellular means there's only one copy of the DNA. I suppose you could argue that, but there's probably better ones to say. Yeah, like circular versus linear. I like that one. Circular versus linear. Histones, yeah, only eukaryotic DNA has histones. Very good. You could put that one. The circular and linear one, basically what that means is if you straightened out all the DNA in a prokaryote, it would form a continuous loop, like a circle. Whereas if you straightened out the DNA in a eukaryote, it would have a start and an end point. So yeah, that's what we call linear. And then yeah, histones is good. Let's just put that as a bullet point. Histones. Histones are just the little proteins that the DNA wraps around. So it helps to like fold it up. Cool, nice. Then next question, what is the internal membrane of a chloroplast called? It's a little bit confusingly worded. You might be like, do they have an internal membrane? Basically, what is the structure in the middle of a chloroplast? It's like made of membrane, hence the question. Yeah, nice. The thylakoid. So thylakoid membrane. They're kind of like in these stacks of discs. You might see them something. Actually, I'm trying to draw a 3D image in 2D. It's not working very well. But basically, yeah, there's stacks of discs. They have the connections called lamellae between them. And yeah, this is the membrane where all the proteins for the light-dependent reaction they're embedded on. So that's where that all goes down. Nice. Cool. Then next one, which enzymes are involved in DNA replication? So there's, well, all specs, you need to have at least 2 for this. Some specs, you also need to have a third one as well. So let's see what you guys can come up with. Oh yeah, someone mentioned granum. So collectively, this is known as a granum. There's lots of thylakoids together. Granum. So I suppose, yeah, technically you could get away with that. I would say thylakoid is better, but granum would be okay as well. Yeah, so nice. Looks like lots of people got these correct. So the ones that you all need to know, you got DNA polymerase, and you got helicase. Sometimes called DNA helicase, but you actually don't need to put that. Put capital. So DNA polymerase, that's the one that joins up the nucleotides in the newly forming strand. So that's catalyzing the formation of the phosphodiester bonds. Helicase is the one that's breaking the hydrogen bonds between the strands. And then some specs, I think definitely OCR need this one, and maybe one of the EdXL specs. But ligase, this is to do with joining sections together on the lagging strand. The sections are called Okazaki fragments, out of interest. But not all specs do that, so we won't go into full detail on that. But if you recognize this, then chances are you probably need to know that it's in your syllabus. The same enzyme comes up in the genetic engineering topic as well, so you might recognize it from that. Cool. And then, yeah, which direction does DNA polymerase work in? And yeah, and why? What can you guys come up for that? Ligase is not included in the AQA syllabus for DNA replication, but the other two are. Out of interest, what specs are you guys studying? Is everyone A level, or have we got some other like IB or other stuff in there as well? Got some, one AQA, nice. To be fair, if you're doing IB, it's also very relevant for that. Even if you're doing IGCSE or like a normal GCSE, the stuff you need to know for mitosis for GCSE actually isn't too dissimilar to A level. Obviously, there's a little bit more detail at A level, but this will still be helpful, even if you're a GCSE student. Cool, yeah. Seems to be a mixture of AQA and EdXL. AQA is often the most common. That seems to be the one that like the largest number of students sit each year. Yeah, nice. 3' to 5' direction. We can represent the word prime just with a little apostrophe, or you can write it out if you want. And the reason why is because it's an enzyme. It can only bind to a specific shape substrate, and the DNA is differently shaped on either end, so it can't bind to the 5' end. DNA runs anti-parallel, and this is why when you're replicating the strands, you have a leading strand and a lagging strand to do with how quickly they can be replicated. But yeah, that's a whole different session. We won't go into that fully today. Yeah, and then what do you guys know about mitosis already? What can you remember? Maybe some like keywords or a interesting fact or something like that. Nice. Prophase, anaphase, yeah. Lots of you guys got mocks coming up. Yeah, fair enough. I think February is quite a common time to do them, isn't it? Yeah, like straight after the half term, a lot of people are going to be sitting their mocks. But yeah, I don't think it's like cause to stress. Like this is still a long way before the exams, and that's why you do the mocks now. It's just a useful way for you guys to get a benchmark, so that you know where, like, what your current level is and how far you like, how much work you need to do to be hitting your like sort of target grade. So yeah, I don't think you shouldn't be stressed. A little bit of nerves is always good. I think it, like, the adrenaline helps your brain actually work a bit better. But yeah, definitely not like really stressed. Spindle fibers, nice. Binary fission, yeah. Nuclear division, very good. Cytokinesis, yeah, perfect. These are good keywords. Nice, yeah, and we'll go through some of these. That sounds like you guys already know a fair bit. I actually find being a little bit nervous for an exam, it also helps with the revision, or it helps with the motivation. You need like a bit of the fear to get you motivated. Sometimes it can be useful. So use it as a tool. So this is the page I was talking about. So we're gonna look at the steps, we're gonna look at the roles, and we're gonna look at the gap stages in interphase, and we're gonna look at binary fission. Mitotic index is something you guys need to do. It's one of the required practicals, but this is a whole other class, so we won't look at that today. These are the specs. You should all have a copy of your syllabus anyway. If not, just download one. You can just type it into Google, comes up pretty quickly. To be honest, for this topic, they're all fairly similar. So yeah, it would be pretty standard for everybody. Cool. So let's recap some of the basics. So this is a representation of a chromosome. A chromosome is just a section of DNA which contains multiple genes. So it's like a tightly coiled section of DNA. The genes would be just like one bit. So maybe from here to here could be a gene. And we call the position of a gene on the chromosome the loci. Well, actually, I guess that's plural. So it'll be locus for a singular one. And there'll be lots of genes contained within a chromosome. So before you undergo mitosis, you need to replicate the DNA. And this takes place in a stage called interphase. And the reason you need to replicate the DNA is because you're making two new cells. You're splitting one cell into two cells, and each of those new cells needs to have its own copy of the DNA. So before you divide, you replicate. And then you get these characteristic X-shaped chromosomes. So whenever you see an X-shaped chromosome, you know that the DNA has been replicated. Because the X shape is because you've got two copies of the chromosome attached together. It's almost like you've got a paper, you photocopy that paper and you staple it together. Now you've got two identical copies. And there's a lot of terminology you need here, and there's a lot of similar words which people often get confused. So let's just go through some of the things we're looking at. So these are both chromosomes. They're attached together at a centromere. That's just the point which they're joined together. And when they're attached together, we refer to them as sister chromatids, which essentially just means identical copies of the same chromosome. So yeah, sister chromatids are like this. People always get this term confused with homologous chromosomes. So a homologous chromosome is the other version of that chromosome. So I'm just quickly drawing one out here in the same style. So these are sister chromatids. These are also sister chromatids. I'm not supposed to write it, but sister crow. And because you've got two copies of each chromosome, I'll draw this one here as well before replication. So this is one copy. Let's say this is the maternal one. Maternal in red, and then the paternal one is in black. You have two versions of each chromosome, and this is because you inherit one from your mother and you inherit one from your father. This is why cells are diploid, which is just another way of saying that they have two copies of each chromosome, or two versions of each chromosome, I should say. So the different versions, the maternal and the paternal, these are what's known as homologous chromosomes. And it's the same for these. These are also homologous chromosomes. So sister chromatids, identical copies. They have the same genes at the same loci, and they have the same alleles. Homologous chromosomes, they have the same genes at the same loci, but they're going to have different alleles because they came from different sources. So the maternal ones, obviously from your mother, the paternal ones from your father. So they're going to have different alleles. So these are not identical, they're homologous. Thinking of like the the genes, loci, and alleles, that's a really good way to remember the difference. So sister chromatids, same alleles at the same loci. Homologous chromosomes, same genes at the same loci, but different alleles. Yeah, so hopefully that makes sense. If you're thinking like, what is an allele? Yeah, basically just an allele is a version of a gene. And you'll learn a bit more about that when you do the A2 if you're an AS. Cool, yeah, so hopefully that makes sense. And let's go through the phases quickly. I'm going to throw these out as questions. So we're in prophase. This is the first stage of mitosis. Can you guys tell me what's going on? There's a few prompts. So what's happening to the chromosomes? What's happening to the centrioles? And what's happening to the nuclear envelope? And we'll just jot down those things. So we'll see what you guys can come up with. Yeah, chromosomes condense. That's the best word to use. We'll just quickly define that. This means like what should we put? Shortens. Shorten, thicken, plus become visible. All of those things are sort of implied by when you say chromosomes condense. So this is the best keyword to use. Centrioles, yes, they move to opposite poles. That one rhymes, which is kind of nice. Helps you to remember it. Centrioles move to the poles. The poles are either side of the cell. So think of it like a globe. You've got the poles, the North Pole and the South Pole. Doesn't matter which around it is. So these are the poles. Obviously, they don't really have a north and a south in a cell. It's just the same word. These are the poles. And then down the middle, we have this imaginary line called the equator, which is the same as a globe. Obviously, there's no such thing as the equator line in reality. It's just like a hypothetical thing we draw to show the halfway point. The equator. Have any of you guys ever been to one of those places where you can like stand on the equator? It would be like, I don't know, that in like, well, it goes all the way through Africa and through Southeast Asia and South America. There's different places you can go, and you can stand with like one leg in the northern hemisphere and one leg in the southern hemisphere. But obviously, it's all like there's nothing actually there. Anyway, yeah, well, it's kind of cool, but not that relevant. Then yes, nice. Nuclear envelope disintegrates. Or you can say like breaks down, disappears, something like that. This isn't a particular keyword. It's just like the meaning. So you could use different things here, but this one is actually a keyword. So I would definitely go for condense. Cool, nice. Let's go to the next one. So metaphase, out of interest. Prophase, metaphase, anaphase, telophase, and it starts with interphase. This is actually not part of mitosis though. So these ones are mitosis. And interphase is first. So one thing that some students find helpful to remember the order is like a mnemonic. You can use these quite often in biology. They can be helpful. So the mnemonic for this is, well, one of the ones. This is the one I know. Maybe you guys know the ones. So I, P, more after T is a way you can like help to remember the stages and the order. Maybe you guys already know them. So maybe that's not that helpful. But if you struggle, that can be a good way to remember them. So yeah, spindle fibers start to form. Nice. Start to form. We could put like from the centrioles. So they're growing from the centrioles at the poles, and they're starting to sort of pull the chromosomes around. So the spindle fibers start from the centrioles and attach to the chromosomes at the centromere. So they're attached now here and here, and that's what's actually maneuvering the chromosomes around. And then chromosomes are lined up along the equator. And again, that's that like hypothetical line down the middle of the cell. So perfect, yeah, they're lined up. And yeah, let's move on to the next phase. So anaphase. What's happening to the spindles? What happens to the centromeres? And then, well, that's kind of like there already. So yeah, nice. Spindle fibers contract and shorten, we could put. This pulls apart the centromeres. So centromeres split and sister chromatids are separated. So now one sister chromatid is going to this pole and the other pole. They're being dragged to opposite poles. And because they're being dragged by the centromere, you get this very characteristic V-shaped chromosome. So whenever you see a V-shaped chromosome, you know that that cell is in anaphase because the chromosome is being actively pulled. So that's a good thing you can put. That's also a good one to put for mark schemes. Like, how do you know that cell's in anaphase? Because it's got V-shaped chromosomes. One thing that causes a lot of confusion is like, when do you refer to them as chromatids and when do you refer to them as chromosomes? So when they're attached together, you call this a chromosome rather than like two chromosomes because it's only one centromere. So you count the number of chromosomes by the number of centromeres. So this is a chromosome. This is also a chromosome. This is a chromosome that's made up of two sister chromatids attached at a centromere. So it's like your photocopy stapled to your original copy. It's still called one chromosome though, while they're attached together. However, as soon as you split them, so this is going to be split apart. So this is a chromosome made up of two sister chromatids. As soon as you split them, you can call them chromosomes again. So now that they're separated, we've got two chromosomes. So once they're detached, call them chromosomes. While they're attached together, you can call them sister chromatids. Hopefully that makes sense. They're usually not too strict on this, but I have seen mark schemes where they wanted it exactly that way around and people lost marks basically. So do just be careful with that. I would recommend you like just follow that naming pattern. V-shaped chromosomes formed, and this is characteristic of anaphase. Cool. So, yeah, I would actually say, yeah, so this is kind of like what we we're just talking about. So I wouldn't actually refer to them as chromatids anymore. I would now say chromosomes. So let's change that. Chromosomes reach opposite poles. And then next, a nuclear envelope reforms. And yeah, after this, we're going to have cytokinesis. Which again isn't technically part of mitosis. It comes after mitosis. And this is division of cytoplasm. And then obviously also division of the organelles that are contained within the cytoplasm. That's also going on. Nice. So yeah, hopefully that all makes sense. Out of interest, yeah, how are you guys feeling with regards to the stages? 1, 2, or 3? Or you can just do a like an emoji instead. Cool, nice. Some ones. Dara, you're like a bit neutral. Hopefully once we do a few more questions, we can like pull you up to a 1. Yeah, nice. So let's have a look at this. So this shows a cell from onion tissue. The student concluded the cell in the image above was in the anaphase stage of mitosis. Was she correct? Give 2 reasons for your answer. And it's a 2-mark question. So 1 mark for each answer. So how do you know that cell is in anaphase? What would you say? Yeah, so chromosomes are opposite poles. Move to opposite poles. Nice. And how else do we know? Yeah, V-shaped chromosomes. Nice. That's a good one to put. V-shaped chromosomes. So yeah, very characteristic V-shaped. And we can see lots of them here. This is because they're being pulled by the spindle. Perfect. It can actually be a little bit tricky to tell the difference between like late anaphase and early telophase. I think the best way to tell the difference is if you can still see a V, then say anaphase. If you can't see any V's, then say telophase. But obviously there's like, when it's very late one, very early other one, it can be a little bit tricky to tell. Okay, nice. Let's look at the cell cycle. So we just looked at mitosis, which is a stage, which was made up of the 4 bits, pro, meta, ana, and telophase. And cytokinesis, cell division, the division of the cytoplasm, comes after mitosis. And the whole rest of the time, we're in this thing called interphase. So this actually is the majority of the cell cycle. Yeah, a figure that's sometimes thrown around is 90% of the time. This wildly varies. Some cells, maybe it's less. Some cells, it can be a lot more. Like some cells actually never leave interphase. If they're not going to divide anymore, they can stay in interphase kind of indefinitely for like years. So yeah, usually like minimum 90% of the time. So interphase is important because during this time, during interphase, cell carries out normal functions. So the cell can't really function properly when it's undergoing mitosis. So it functions normally during interphase. And if it's getting ready to enter mitosis, it does several important things during interphase, which get ready for cell division. And we'll look at what those are on the next page. So this is mitosis. And then the other bit said, this is all interphase. Mitosis, interphase. So technically, mitosis actually doesn't mean cell division. It actually just means division of the nuclear material. Division of nucleus. Basically, division of the DNA. And let's go through the different phases of interphase. So there's 3 bits. G1, which some specs call growth, some specs call gap, or you can just refer to it as G. If you don't really want to use either of those, it's fine, just call it G1. Let's put G1. The synthesis stage, or this is also known as the S phase, and you've got growth or gap 2, also known as just G2. So what do you guys remember about these different phases? What goes on in G1, what goes on in S, and what goes on in G2? And we'll jot those down on this page as well. Yeah, everyone always goes for that one first. So synthesis stands for synthesis of DNA. So DNA is replicated. Nice. And what about the G phases? Yeah, you will need a lot of ATP for the spindle fiber contraction. So definitely mitochondria are going to be involved. Nice. Yeah, so in both of these, hence the name growth, the cell grows. To be honest, actually, these are both the same for most of them. So cell grows, protein synthesis. So you can't do protein synthesis in S phase because you're replicating DNA, and you can't do protein synthesis in mitosis because the chromosomes are condensed. So you can only do it in G2 and G1. Yeah, nice. And then kind of run out of space here. So remember, these are the same. I'll put it down here. Organelles replicate. And this makes sense because both of the new cells need to have copies of the organelles as well. So all the mitochondria, ER, lysosomes, they all replicate as well. Then there's an extra step in G2 because we've done the we replicated the DNA. You also, there's a checkpoint where DNA is sort of like proof read. It's checked for errors. So this is just like an important step to make sure that DNA replication has happened correctly. The DNA polymerase is actually very good at replicating DNA. Its mistake rate is like way better than any human. I think it's like a couple of bases in every billion it would get wrong, which is like, yeah. If you ever tried to type out, you know, those, sometimes you have to put a Wi-Fi password in. That's one of those like awkward ones where it's like different letters, uppercase, lowercase numbers. It can be tricky just to write those 9 digits down correctly. So yeah, this is like making only a mistake every like billion characters or so. So pretty good, but it does still make mistakes. So it's important to check it for errors. What's the name of a mistake in when the base sequence is copied incorrectly? What would that be called? And why is that bad? Oh, actually, I can see someone's mentioned this word already. Alia, you put it. Yeah. So we'd call that a mutation. Put it on the page as well. Check for errors. This is a mutation. And mutations, obviously, yeah, they're generally bad. Occasionally you get a good mutation. That's how we evolve. So mutations are actually an essential part of natural selection. We wouldn't be able to evolve without mutations. However, you don't really want to be the one that's rolling the dice and taking the gamble on the mutation, because the vast majority are disadvantageous. So best to play safe and just try to have no mutations. Yeah, it leads to non-functional proteins. It can lead to cancer as well. Yeah, exactly. Nice. So cool. This is a really common type of question you might see, where they show you a graph like this. So on the x-axis, you've got time. On the y-axis, you're going to have something like mass of DNA, or it could be something different, but yeah, like amount of DNA, something like that. Then you got different stages. We can see the DNA mass is changing. It's increasing here, it's decreasing, it's staying the same, increasing, decreasing. And then you can be asked to name different bits. So can you guys recognize like what's happening at A, what's happening at B, what's happening at C, and what's happening at D? And how do you know? Or I should say, like, not what's happening, like which phase of the cell cycle are we in? So you can just give like mitosis G1, S, G2. Yeah, B to C is S phase. So in fact, I'm going to change this a bit. So here to here, this is the S phase. We know that because the mass of the DNA has doubled. G1 is actually labeled. So we know this is G1. So G1, S, and this is also G1 here. G1, S phase, something, back to G1. So we know that somewhere between C and D, we've got G2, and we've got mitosis. We actually, from this graph, we can't tell when G2 finishes and mitosis starts because the mass of the DNA doesn't change. The only thing we know is when it suddenly halves. So at D, this is cytokinesis. So when the two cells finally separate, each has just one set of the DNA. So rather than having double, 5 rather than like 2.5, it straight away drops back down to half. So yeah, that's very like obviously cytokinesis. So yeah, they can ask questions different ways on these graphs, but as long as you can work out where you are, and usually it's by looking at S phase and cytokinesis, then you can figure out what's going on. Nice, yeah. So hopefully you guys are all still happy with that. And yeah, let's try some more questions. So we've got a diagram here. Take a second to have a read of the context, have a look at the diagram, and then the question is explain why some cells have a mass of DNA between 1 and 2. So take your time, have a read, and we'll see what you guys can come up with. So yeah, why are they between? So the majority have one set. The second most is a double set. And there's some which are in between. Yeah. Yeah, you got the right idea, Alia. So these ones must be in S phase. They are undergoing DNA replication. So they've got more than one set of DNA, but they don't quite have a double set. So they're actively replicating it. Oh, N15, yeah, like the Mitosis and Stahl experiment. Yeah, good guess. That's not what they were getting at here, but that is actually an important thing that they use to identify the semi-conservative replication. And it's a cool experiment. It's kind of like, it's relatively simple, but demonstrates the point really neatly. Those guys, they won a Nobel Prize for it because it was just a good bit of science. Using the limited technology that they had back in the day, they were able to prove this. So anyway, next question, bit of maths now. So a cell cycle takes 4 hours, 18 minutes. How many minutes to make 8 cells from one cell? I'll give you guys a little time to have a think, and then we'll go through this. And our answer is going to be in minutes. Anyone want to throw out an answer, or are you still working it out? It could be guys a little bit longer, and we'll go through the method. So we've got some answers. I don't think either of those are quite right. So let's go through. Although I haven't actually done the maths yet, but I think it's in the 700s from what I remember from doing this question a few months ago. But let's go through the method. So yeah, I guess let's firstly work out how many minutes are in one cell cycle. So basically, what is 4 hours and 18 minutes in minutes? So to do that, 4 times 60 plus 18. So let's quickly work that out. I'll have a go as well. I'm just using my phone, but it should be fine for this sort of maths. So that's 258. Actually, I think about that. Probably could and should have done that in my head. Then, so that's one cell cycle. and we need to know how long it's going to take to go from 1 cell to 8 cells. So you can kind of, because it's not that many, you could actually just draw out a diagram if you wanted. 1 cell divides once, gives us 2 cells. This divides again, gives us 4 cells. And these divide again. It's going to give us the 8 cells that we need. 1, 2, 3, 4, 5, 6, 7, 8. So this is 1 division, 2 divisions, 3 divisions. So we just see 3 times the length of time for 1 cell cycle. So 3 times 2, 5, 8 equals 7, 7, 4. Yeah, nice. And that's our answer. So it's 3 divisions. That was a key thing to work out. What you could do, because there's not very many, you could draw it out or just literally like work it out in your head. If they said like, we want to make like 8,000 cells or 8 million cells, then you can't really just draw that out. That's going to take too long. It's not going to be practical. So there's actually a shortcut you can do. So, or you can use it to test it. So the number of divisions is equal to 2 to the power of how many divisions you had, because 2 to the power of something just means 2 times 2, however many times you're going to do it. And cell division is it's timesing by 2, they're doubling. So from 1 cell to 2 is timesing 2, 2 cells to 4 times by 2, 4 cells to 8 times by 2. So we can represent this mathematically as 2 times 2 times 2, which is the same as 2 to the power of 3. So this is a useful way of working out how many cells you've got from cell divisions. So you can just square root 8 to get the number of cell division this time. Cool, nice. Hopefully that makes sense. Then, yeah, let's have a quick look. How are we doing for time? Got like 5 minutes left. That should be plenty to just do this. So the final thing I want to do today is just look at binary fission. This is basically the way in which bacteria divide. And there's not really a huge amount you need to know for this. The key points are that they do DNA replication. So circular DNA replicates. And kind of similar to eukaryotic, each of the loops goes to one end of the cell. DNA loops move to opposite poles. Plasmids, which are the smaller loops of DNA, these also replicate. And again, move to opposite poles. Move to poles. Then eventually the cytoplasm just divides between the two. And now you've gone from 1 bacteria to 2 bacteria. Divides down the middle. And the cell wall is basically completed. So they build a cell wall all the way along here, all the way along here, until we get our 2 finished bacteria. This is quite quick. So depending on the bacteria, the generation time is different. But for something like, let's say like E. coli, the generation time can be anywhere from 20 minutes to about 1 hour. So they're pretty quick. They can they can double roughly, well, at least once an hour. So this is how bacteria can, like, they can reach very large numbers very quickly. If you're doubling every 30 minutes, you're growing exponentially. You can go from a small number to a huge number in a very short space of time. So there's some crazy figure, like if you start 1 bacteria, 1 E. coli, and you gave it a week of unrestricted growth. So you start with 1, 20 minutes later, you've got 2, 20 minutes later, you've got 4, and so on. If you left that for a week and none of the bacteria died, the mass of those bacteria would weigh more than the whole of the universe. So kind of crazy. That's how big the numbers get when you have exponential growth. The reason we're not like inundated with bacteria and this doesn't happen is because they also have a short lifespan. So they can replicate quickly, but they also die quickly. So yeah, we don't end up with like huge numbers because they run out of resources, they die, and so on. They're actual, like, even if there was enough resources, the cells don't last that long. Yeah, nice. So yeah, how is everyone feeling with that? The binary fission, I think, actually isn't in all specs as well. So if you've never heard of that before, chances are you don't need to do it. I think it's not in all of them. Cool, nice. Thanks, Rennie. Yeah, perfect. So yeah, hopefully you guys are good with that. You can tell the sages, you can look what's going on in interphase, you can interpret graphs, and you can discuss binary fission if you need to know. Yeah, nice. So I think we will end there, a couple of minutes ahead of schedule. So that's good. Yeah, like I said, we do these sessions on Sundays. We do one for each subject every month. So the next biology one is coming up at some point. If you want to set a reminder, you can go to our YouTube channel and you can set a reminder through YouTube. I think they send an email notification. I've actually never done it, but I imagine that's how it works. Or maybe you can link it to something other than email. Yeah, so fill it out. They have subjects as well. If you do maths, chemistry, physics, join on one of those sessions. We've got tutors who are doing those as well. Or if you enjoyed the session today, you can go to our website and we have like a lot more of these classes on our own website. It's like a subscription model. But yeah, we're doing more on there. So you can also check that out and have a look at those. Cool. And yeah, I think that's it. Thanks for joining as ever. Good luck if you've got a mock coming up. I hope it goes well. Yeah, well, hopefully it goes well. If it doesn't, don't overly stress. It's still early days. You've got time to make changes, and it could actually just be the wake up call that you need if they don't go too well. So yeah, touch wood, they are good. If not, you've got time to turn it around still. So don't be too down. Yeah, nice. All right, thanks guys. See you next time. Bye. Bye.